So thanks for coming today, this morning early for this talk. I'm gonna try to make it a little bit more translational rather than as basic as possible. So hopefully keep people interested. Little passion of mine in terms of retroviruses, which have kind of been a recurring theme in my life. And so I kind of meshed, my interest for retroviruses and glioblastoma. And I'll kind of talk a little bit about how the two intertwine in terms of, an etiology of glioblastoma, and then also using retroviruses as a treatment for glioblastoma. So on two sides of the coin, hope this will kind of inspire some of the residents um, and students to kind of be interested in kind of scientific pursuits. And coming from someone who wasn't a scientist by training, I think this is really been motivating me a lot. So, I show this slide at every talk. We know that, recurrence in glioblastoma is inevitable. Prognosis has been extremely poor. You see that curve there, that Kaplan-Meier is, well known to every resident. For 20 years, this Kaplan-Meier curve, we've been staring at it and hasn't really moved much. And, mostly because, this has almost been a little bit of a hindrance in itself that we have this protocol that we aren't able to kind of move away from it. And, we know outcomes aren't that great, and recurrence is really inevitable. And so, I put a picture of Timur here. I wish he was here. But this was, I see him all the time in the physician dining room, and this is him signing his contract. He said, what are you guys doing with glioblastoma research? It's basically throwing your money down the toilet. You're just basically doing this 20 years research. You guys haven't done a single thing. But in spine surgery, they come up with the different screws and radiolucent screws and new rods and minimally invasive techniques and AR and this and that. And I haven't been able to show like a Kaplan-Meier curve for spine surgery, but our curves haven't changed much. So he kind of got me thinking. I was like, well, what are we really doing to move the field forward, and have we really made a dent in outcomes at all? And if you look at funding for glioblastoma or brain cancer, it's been pretty poor over the last decade or so. If you look at breast cancer, prostate cancer research, the funding is quite high. And glioblastoma is considered this orphan kind of disease, because it affects few people, but it is the most common primary cancer of the brain. And you look at the outcomes. I mean, there really hasn't been much changing. Like so, 1977, 1980, radiotherapy, and then the Stu protocol in 2005, and in 2009. We had Avastin. That was like the big, kind of update for improving progression-free survival. And since then, really not much has really moved anything. Maybe tumor treatment fields, but not much. So the question everybody asks, what are we doing differently? And the current paradigm is kind of made the same. We take a patient to get imaged in the ER, you take them to surgery, you check for molecular testing, you do radiation and chemotherapy, and we tell the patient the same thing. We're going to test for different molecular targets, and hopefully we're going to find a treatment for you. And most of the time there's no molecular target, and they get the standard of care, and then treatment occurs. And so there's a few subset of patients that we have targeted treatment for, but most of them there's not. So I think there's been a big push in the literature kind of using multi-omics to kind of, figure out the drivers of these tumors. I was just talking to Dr. Raga, Antonio Ivarone, he's come now as a deputy director of the Cancer Center, and he's kind of focused a lot on this kind of approach to figure out what are the master drivers of brain tumors. And, can we target these master drivers as, to figure out, the cell of origin and target them and improve outcomes. From my perspective, I'm more interested in viruses, and, the cancer virology has been a really kind of interesting field that's really developed over the last, 200, 300 years. And the WHO estimates that 10% of cancers are related to viruses. I think there's probably a little bit more than that, but at least they've been able to identify viral DNA in tumor specimens. This is really known. A very popular virus has been documented in most GBM patients. Do you guys know which virus that is? That they've found, remnants of these viral particles in GBM. Common one causes sore throat. CMV, yeah. So CMV has been, they've been finding, relics of CMV drivers in GBM. And so there's a whole discussion maybe 10, 15 years ago, whether or not this was the etiology of glioblastoma. And, since then they've kind of proven that wrong, but that was a big talk. There's also, some other viruses I'll talk about as well. We know that viruses can elicit oncogene expression, and they can also cause host gene modification. So this has been a really kind of important topic, and in glioblastoma, it hasn't really been investigated too much. I always show this this slide. This is Peyton Rose. I'm not really good at the French here, but essentially, so this is like in the early 1900s, and here he's looking at basically chicken sarcoma virus. And so what he was doing was he realized that he could basically induce chicken tumors by taking a tumor from a chicken, digesting it in a blender, running it through a filter, treating it with antibiotics, and then taking whatever that remnant solution is, that it's cell-free, that has nothing else other than the supposed DNA or RNA, whatever this is, and puts it back into another chicken, and then they develop tumors. He said, well, how is this possible? There's no cells in here, there's no proteins in here, there's nothing in here other than, you know, this antibiotic treated solution. And so he had this hypothesis that there's some sort of virus in here that causes cancer. He didn't know what it was and kind of ignored it. And that was in the 1910, 1920. 50 years later, it wasn't really David Baltimore, Ronaldo Dubelko. You guys probably know him from the media. Dubelko's DMEM solution, and Howard Temin kind of got the Nobel Prize for really discovering really what was in this this fluid. And what it was an RNA tumor virus, and they realized that on subsequent passages of the cells that they still retained the same initiating, they still had the same drivers, you know, and these drivers were not diluted over time, which means that somehow that RNA that was in that chicken tumor was integrated now into the DNA. And so that's impossible because the central dogma, which we were all kind of taught in elementary school, was that you have to go from DNA to RNA to protein. But for some reason, this virus was going from RNA to DNA and integrating itself in the genome and into our germline. So this is kind of a very big discovery. And at the time, this was going on in the 1980s. There was a first time, there's a pandemic going on with a virus in humans, affecting a very kind of a very unique population, and it was very scary. Nobody knew what this virus was. Nobody knew how it worked. And they actually realized that this was actually a retrovirus, and it was able to integrate itself into the DNA of its host. And so this was, you know, against the central dogma. They discovered that this was mediated by reverse transcriptase, and this was a kind of a huge discovery. What's interesting is most retroviruses actually are oncogenic, as in they do contribute to tumor formation, and AIDS is no different. So then the question that you ask yourself is what viruses are really overexpressed in GBM? And we talked a little bit about CMV, but this was a paper published about, you know, 6 years ago in Clinical Cancer Research, where they basically showed that there's really two main viruses that were kind of associated with the worsening prognosis. One of them is woolly monkey sarcoma virus, which, I mean, is a very rare kind of virus, but they were able to find some evidence of infectivity in patients with glioblastoma. And interesting, this one has a oncogenic transgene. So this was associated with worsened survival in patients. They had about 190 patients here in this sample. But interestingly, if you look, you know, and they did, you know, they looked at, you know, about 30 or 40 different viruses, and, you know, this one human endogenous retrovirus K was positive in about 280 samples. And so that's like pretty high positivity rate. But, you know, really, what is this virus and what does it do? And is it really relevant in glioblastoma? So, you know, the name speaks for itself. It's a human endogenous, meaning it's in our genome, and it's a retrovirus. So this forms about 8% of our human genome. So, you know, if you look at any one of us, if you take 8% of our genome, that basically is formed by retroviral elements that we acquired while we were evolving. So sometime when we were monkeys and evolving to humans, we've acquired these germline infections over time, and they've been helpful. You know, these are, you know, evolutionary gains that are in our genome that help us evolve. But our bodies kind of found a way to suppress these viruses over time, and they've added mutations and deletions. So essentially, we're not out here, you know, forming retroviral particles in our normal day living, because our bodies epigenetically suppress them. But in cancer, sometimes this can get reactivated. So this is, you can actually find a few areas in our genome. I think there are about 10 areas in our genome where you can find full length retrovirus transcripts. This is quite interesting, and it becomes important for several reasons. And when I say 8% of our genome is formed by retroviruses, you ask yourself, well, this is just junk DNA, right? Nobody really cares about junk DNA. They don't do anything. But if you think about it, 2% of our genes encode proteins. So 98% of our genes really don't do anything. And 8% of those are formed by retroviral elements, and 60% of those are repeat DNAs. And so there's a huge, conversation about what is this dark genome? What is it doing? Is it doing anything in cancers? Is there a reason we should target it? It certainly, encodes for more than, just the 2%. And when you look at conventional data sets like TCGA, everything we use for cancer, screening and things like that are all ignored. They only focus on this 2%. And so you actually ignore, 98% of the genome because you think it's junk DNA. And I'm going to hopefully convince you that, this is not the case. So these HERV proteins and transcripts are actually overexpressed, in the most stem-like state. So if everybody remembers, I'm sure Dr. Raghav knows this, but the syncytial trophoblast and embryogenesis, this is a very important protein is a syncytin-1, and it's necessary for development of cell-to-cell fusion in embryogenesis. And when you eliminate this protein, the syncytial trophoblast does not form. And this is a retroviral envelope protein. So its homology is very similar to, the HIV envelope protein or other retroviral proteins. So this is the first kind of important functional, retroviral protein. And there's so many more that are kind of ignored as well. And so if you think about the most stem-like state of our human in our body, it's the blastocyst, the 8-cell stage, when we're kind of, all the cells are pluripotent. And this is an important paper that came out in Nature in 2013. And basically what they showed was that in the blastocyst state, and if you do electron microscopy, you can actually see viral-like particles being expressed from the blastocyst, meaning that you can see, retroviral proteins on the surface. And you can see this on the dark here, where it's basically labeled VLP. You can see them being expressed in this state. And as they differentiate, and we become, more selected, these proteins actually disappear and are epigenetically silenced through DNA methylation. And so, this is the kind of a interesting concept, because you think about it, cancer and these stem-like states when we're in embryogenesis are very, very similar. And so this got us to thinking, that these endogenous retroviruses could be expressed in a variety of cancers. And I show this picture of a koala because the story of the koala is very interesting. I don't know if anybody's been to Australia and seen some koalas in Australia. Oh, Victor, Lou, of course. So the koalas in Australia are very interesting because they are undergoing active endogenization of a retrovirus currently. So the northern areas of koalas are actually getting a retrovirus inserted in their genome currently, and so as they are procreating, they're spreading this retrovirus along their germ line. And the southern koalas don't have this in their genome yet. And if you look at the cancer rates in the northern koalas versus the southern koalas, there's a much higher rate of cancers in the northern koalas. And when you look at the sequencing, you can see that this retrovirus that they call a koala endogenous retrovirus, a KORV, is actually inserting itself into the areas which are tumor suppressor areas or oncogenes. And so they're driving tumor formation in koalas in Australia. So in real time, we're kind of visualizing this happening in another species. And so you can imagine this was kind of going on in humans 6 to 10 million years ago. And so we kind of wrote this paper kind of discussing all the different ways that these retrovirus proteins can kind of cause cancer formation. They can go and deactivate tumor suppressors. The proteins themselves can activate tumor activating areas. They can insert themselves and cause insertional mutagenesis. And there's been quite a bit written on this in other types of cancers, but in gliomas, really nothing has been written about this, other than kind of one small paper where they looked at, RNAs for this protein. So we kind of started off with an exploratory analysis of HERV expression in glioblastoma. A lot of thanks to Vaidya for doing this preliminary work, kind of demonstrating that endogenous retroviruses are overexpressed in glioblastoma. Here, we were able to show that one of these endogenous retroviruses in chromosome 19 were much higher expressed in glioblastoma compared to astrocytes. And this was kind of a retrovirus that's kind of been really ignored in a lot of literature. And we actually made a antibody to this protein, using a custom antibody, and you can see this much higher expressed in GBM than in epilepsy patients. So here on the left, you can see the epilepsy, IHC. This is glioblastoma, much higher expression. This is RNA in situ hybridization. So now we're, basically, these red dots are every RNA molecule that is this endogenous retrovirus, and you can see survival. You have higher expression of this transcript. The patients do much worse compared to patients who have low expression. And when you look at the subtypes, you can see the classical GBMs have the highest expression, and as expected, the IDH mutants, which are highly epigenetically, silenced, a lot of DNA methylation, have lower expression of endogenous retroviruses. So this was published, last year, and we kind of started to expand on this work a little bit more because we thought that we weren't capturing all the endogenous retroviruses using this portfolio. So we kind of used a bigger data set. We took about 75 patients from Miami and kind of did deep sequencing on them, and then kind of tried to figure out whether or not which ones are kind of driving glioblastoma. And this kind of led to this paper, which was published in July of this year, kind of suggesting that this specific endogenous retrovirus K, which is the most recently integrated retrovirus, this one that's kind of has the full length transcript in our genome, is overexpressed in the stem cell niche in glioblastoma. And so, in order to prove this, you had to do a lot of things. First thing we did was we took a whole bunch of patient samples and stained them for retrovirus expression. So yellow here is the retrovirus here. And you can see that in glioblastoma, it's pretty highly expressed, but not expressed everywhere. Then we took, tumor samples and CSF from patients. So we basically took out a tumor, and then you took the CSF around the tumor prior to surgical resection, and we looked at RNA levels of this retroviral element as well. And you can see, compared to epilepsy patients, they're much higher expressed. In the tumor samples, they're higher expressed as well. And you can see that in astrocytes, there's really no protein expression, and in epilepsy patients, there's really no protein expression here. So this is the retroviral envelope protein, which is considered kind of an oncogenic protein. This is kind of beautiful, colorful heat map. Nobody really knows how to interpret these things, but Vedya is able to kind of work on this a little bit. But essentially, this is kind of looking at every single element. So in our genome, as I mentioned, there's 100 areas in our genome where this retrovirus has inserted itself, and you can see that there's the expression of this retroviral element is very heterogeneous along every single loci. And these are all the different patients in our data set from and what this, as we kind of decode this information a little more, we can see that between normal brain specimens and GBM, this protein is this RNA is overexpressed across the board. So if you look at the overall HML2, it's much higher expressed. The promoters are much higher expressed. The GAG and the reverse transcriptase levels are higher, and this reverse transcriptase will be really important later on, and the envelope RNA as well. And, again, this also correlates to survival detriment in these patients as well. So this is a prognostic factor as well. And so now we're actually using this, hopefully, we're starting a pilot study here at to stain our patients, after surgery for this HERV-K envelope to see whether or not it is a prognosticator for our patients and whether or not it contributes to stemness. And so, you can also see, in the different subtypes that there's, various expressions. So I won't focus too much on that as well. So then the question is, does it really contribute to stemness? I talked about the blastocyst. I talked about how this protein is overexpressed in the most stem-like state. And so what's the role of this HERV-K in glioblastoma stem cell niche? And the stem cell niche is kind of important because those are the cells that are the most resistant to, temozolomide, most resistant to radiation. And those cells that everybody hypothesizes that these are responsible for tumor recurrence. And so if you look here, this is a patient sample. All the sample is all glioblastoma, but in this area here, it's very highly expressed in this HERV-K envelope protein. And if you look at this area, it's a very high expression of SOX2, OCT4, Vimentin, and Nestin. So this kind of got us thinking this is the stem cell rich area of GBM, and this is the area that's most highly expressed with HERV-K envelope. And again, this is recapitulated here in another patient sample. And so, this is great and all. You're kind of showing some slides and showing an area of the slide that's overexpressed, and that's somewhat convincing. But really, the most convincing thing is, can you take a bulk tumor, can you do single cell RNA sequencing and find the most stem-like cells in that bulk tumor and see what are those expressed like? Are those stem-like cells expressing this or not? Are the differentiated cells expressing other proteins or other RNAs? And so this is the real question. So then we took, some sample data sets from Dr. Verhaak as well, and kind of helped. This is about 66,000 cells, and we basically looked at the stem-like cells. This is the red cells. You can actually look at their RNA now for HML2, and you can see that they're much higher expressed in the stem-like cells compared to the differentiated and proliferative subtypes, and specifically in the neural progenitor cells, which are the which are one of the most stem-like cells. So NPC and OPC cells are kind of what they consider as the cell of origin for glioblastoma. So this is really highly expressed in that. Then can you knock this down? So then we took a CRISPR system and kind of knocked down this RNA using CRISPR-Cas9. And you can see the neurosphere formation assays actually come down. You can see there's way more neurospheres than non-guiding. And when you knock it down, they just form spheres less, which is a marker of stemness. You can also look at, the polymerase, the reverse transcriptase levels, and the nestin and OCT4, which are stem cell markers. They also come down dramatically as well when you knock it down. This is, again, kind of, you can show this in one way. You can do qPCR, but you can also show it with immunofluorescence, kind of knocking down, this HERV-K envelope. You're actually reducing stem cell markers with protein. And here, this is RNA at the bottom, nestin, OCT4. We're knocking down expression quite significantly with this CRISPR system. And so then the question is, okay, well, what does this RNA do? So we basically have a plasmid where you can resurrect this retrovirus in its full length form and reintroduce it into cells. This is kind of interesting for several number of reasons. One is that you're kind of forcing activation of this retrovirus, so you can see what it does in cells that really don't express it. And we did RNA sequencing, you can actually see that the cellular programs that are being activated are all cancer related. They're, cell movement, cancer, proliferation of cells, tumorigenesis. These are the activation pathways that are being activated whenever you overexpress this HERV-K transcript. And when you overexpress it in astrocytes, remember I said astrocytes don't express this because they're differentiated. You can actually see OCT4 levels here go way up. And OCT4 is a stem cell marker that's really expressed in the blastocyst and stem-like cells. And you can actually see, and I'll show you this video soon, but you can see that these are 3D cultured astrocytes. They transform quite significantly. So here's a video. I'm going to show it. This is when these are CD34 derived astrocytes. When you transform them with HERV-K, you can see what happens. These are just basically, floating cells in a dish over 72 hours. You give them this retrovirus, and you can see they start forming these satellite colonies. They start proliferating more. Their OCT4 levels go up. So this got us question: is this a tumorigenic retrovirus? I mean, you usually cannot activate and create a tumor in glioblastoma with just doing one gene by itself. And so this is a very interesting thing we found in our paper. And so this got us thinking that this could be a really good target for glioblastoma. And what can we do for targeting? And so when we targeted this in mice, this is a transient knockdown, overall survival improved. And lo and behold, when you look at cells and look at these GBM cells in electron microscopy, you can actually see retroviral particles being formed on the surface of some of these cells. It's a very rare phenomenon, and it's only in the most stem-like cells. So again, pretty interesting discovery to find. This is a control kind of showing you that when you overexpress the consensus plasmid, you actually see the same retrovirus here and the same size retrovirus in naive cells here. So then that's Scott's question, the age-old question: can you use antiretroviral therapy to treat GBM? I mean, if this retrovirus is important, and we know that antiretroviral therapies can affect this thing, this virus, can you use that in GBM? And so we did that, and we were able to show that there is a stepwise decrease in cell viability. This is about 70 or so glioblastoma cells here, showing that most glioblastoma cells are sensitive to this abacavir, which is a reverse transcriptase inhibitor. And when you treat these cells with the reverse transcriptase inhibitor, you actually reduce their stem cell phenotype as well. And then, you can show here again near the bottom, when you treat them with different reverse transcriptase inhibitors. This is work done by Sarah Rivas, who's a second-year medical student, and Jaleesa, who's our postback in our lab, basically showing that proliferation decreases significantly when you treat with these antiretroviral therapies in cell culture. And this is just all the different retroviral, antiretrovirals, and all the different GBM cells. And so it kind of shows heterogeneous effect of these cells. And so can we translate this clinically? These are FDA approved drugs. Can we translate this? Everybody, put your money where your mouth is. Timur is saying that we're wasting a lot of money. So can we actually start a clinical trial, kind of doing antiretroviral therapy for GBM? And so this is a paper that was published in Cancer Discovery, I think. And basically they took patients with colorectal cancer. They showed colorectal cancer has high expression of these repeat elements. So they have a high expression of a lot of these endogenous retroviruses. And they took patients that failed treatment. They failed 3 different types of chemotherapies. And they did single agent lamivudine. Lamivudine is a reverse transcriptase inhibitor used for HIV. They used one single drug, and they had a 40% clinical benefit rate in patients who failed 3 different chemotherapies and radiation for colorectal cancer, this metastatic treatment refractory. And this is kind of ignored, kind of in this, in this journal. I mean, but the very interesting findings. So 9 out of 32 patients had an improvement with a single agent. And if you know anything about HIV treatment, you don't do a single agent, you do multiple agents. And so that got us to thinking that, we can do maybe some drug repositioning of different antiretrovirals for glioblastoma. And so that's kind of got us to thinking about this clinical trial we're starting here in the next few months. It's called Starlight. Basically, we're treating patients with unresectable glioblastoma who are being treated with laser thermal therapy and basically giving them antiretroviral therapy at the same time. We're using 3 different agents. We're using ritonavir, abacavir, and lamivudine to hopefully target the stem cell niche in glioblastoma and kind of potentiate temozolomide and radiation. So these are newly diagnosed patients. We're going to start treatment on presentation, do a biopsy laser, and then basically follow them over the next, until disease recurrence. And so I think it's important that we're doing something new. This is, relatively a cheap clinical trial funded through Sylvester and some industry sponsored funding. But this is kind of a concept that I thought was interesting that you could do quite easily with FDA approved drugs and get something started in patients. And so the next part of my talk is, can we use this to kind of improve, glioblastoma immunotherapy? So there's been a lot of talk about immunotherapy in glioblastoma, and it's failed in a lot of regards in phase 3 trials. And so our lab is also interested in seeing whether or not we can improve outcomes, specifically through retroviral therapy. So this trial, Checkmate 143, is kind of a famous trial that was published, I want to say, 3 or 4 years ago, and their outcomes in phase 3 trials were negative. So they compared nivolumab versus Avastin in glioblastoma, and their outcome was not any improvement. And so the main question here was is the checkpoint inhibitors actually going to the tumor? Are the tumors very cold? Are they kind of immunosuppressive, so they're not expressing these checkpoint cascades on the tumor? And so the question is, can we convert these tumors from a cold suppressive environment to a hot kind of immunogenic environment and then give them checkpoint inhibition? So this is the question: is can we convert these cells and make them more receptive to immunotherapy? And so that's been a big topic in glioblastoma research ever since the initial discoveries that there are some cells that are suppressive, that are immunosuppressive in the GBM tumor microenvironment, and can we target those specifically? And so this got us thinking: can we use viruses to kind of induce immunogenicity in these tumors, and can we induce this expression of these viral RNAs to make the cells more responsive? And so this concept of viral mimicry is related to that. So basically, you basically have these endogenous retroviruses that I mentioned are 8% of our genome. They're normally silenced. And imagine if you take away the silencing. So you take away the silencing of these retroviruses, and you basically have a high expression of double-stranded RNA in the cytoplasm. And what happens when you have a viral infection? What's the main driver there? Is interferon. So interferon levels go high in the cells, and these cells become kind of a warning sign. So this is the idea of viral mimicry: is can you express these double-stranded RNA molecules and induce interferon responses and induce an antiviral immune response that will now potentiate immunotherapy. So this is the opposite. So before we're talking about an oncogenic retrovirus, it's driven by really proteins. Now I'm talking about using the endogenous retroviruses to increase immunogenicity of tumors. And so in order to do this, you can basically take, you can do epigenetic therapies like demethylating agents, for example, like 5-azacitidine, or other kind of genetic therapies. Remove these repressive marks. These viral RNAs then come out and start being expressed, and the cell is not used to this. The cell is not happy with expressing these retroviral elements. So then they start having these interferon stimulating genes, ISGs, and interferon responsive elements that basically start to be expressed higher because of these double-stranded RNAs, and you get high interferon expression and cell death, and you have higher antigenicity. What that means is these cells may be more receptive to checkpoint inhibition. And so this is a paper published by Kate Cipollini and Cell, suggesting that when you inhibit DNA methylation, you get a high interferon response in cancer, and that makes these cells more sensitive to checkpoint inhibition. And this has really been recapitulated in renal cell carcinoma, ovarian cancer, and lung cancer. No one has looked at this much in glioblastoma. So can we do the same viral mimicry, option in glioblastoma, and can we improve checkpoint inhibition in these tumors as well? And so this is funded by the NREF. This is Andy Parsa here in the top right, who basically passed away, but he was able to have this legacy funding, and our lab is lucky to receive some funding for this project. But essentially the idea is, can we unlock endogenous retrovirus expression through these double-stranded RNA molecules and increase interferon responses and increase the checkpoint therapy efficacy? And so specifically, we're targeting this one molecule, NPT20, which is a zinc finger. We consider this the master regulator of endogenous retrovirus expression. And so what we did was we knocked down this protein. You can see here, this is deconvolution through TCGA, and you can see this protein is very negatively correlated to CD8, neutrophil, and dendritic cell infiltration. So this is a very immunosuppressive protein, and so can we target this and in glioblastoma? And here on the right, you can show here that these are all the endogenous retroviruses. They all express themselves in patient samples very, very closely. This is a work done by Christian Ramsumer, who's a MD PhD student in our lab. And so a lot of this expression now is, you can see that this big circles here, these are pretty, but you kind of have to understand what they're saying. So these are the double-stranded RNA sensing pathway. So RIG-I is a double-stranded RNA sensing pathway, and so they're all very co-expressed. And so this is what we're trying to do. We're trying to increase the expression of the double-stranded RNA sensing pathway, and if we induce expression of this, do these cells become more immunogenic? This is the thing I'm trying to hammer home, is that this is a potential way that we can overcome the immunosuppressive GBM microenvironment. And so when we knock down this protein, you get high expression of this RIG-I, which is a double-stranded RNA sensing pathway, higher expression of TLR3, which is the downstream activator there. And I'll show, this is an adherent cells, A172 and U87. So the idea is that you have high expression of RIG-I and low expression of epigenetic repressors. And again, this is recapitulated in the A172 and U87 cell lines. And here you can see the double-stranded RNA. This is a marker for double-stranded RNA. Now you're increasing the double-stranded RNA in the cells when you knock down this protein as well. And so then, that's great, but you have to use an immunocompetent model. So we went to the mouse model, and use an immunocompetent mouse model here. And so we're knocking down this protein, and you can see the PD-L1 expression goes markedly higher in patients in human cell lines, but also in mouse models. And this is a very resistant mouse cell line that is very resistant to checkpoint inhibition. So we wanted to use this cell line because this was kind of a great model for us to target these epigenetic repressors. And if we can improve survival in this cell model, then this makes more sense for humans. So then we use this SP28 model. We knocked out this NPT20. Then we developed a syngeneic orthotopic model, and basically had 4 different groups. We have control plus PD-L1, NPT20 by itself, and then NPT20 plus the PD-L1. And so now we're looking and seeing whether or not outcomes are improved with this checkpoint inhibition and knocking down this protein. You can see in cells, the proliferation rate is attenuated significantly. And this is the most important thing here. See, the PD-L1 expression in the controls are basically absent. When you knock down this protein, PD-L1 goes way high, and the TLR3 activation goes up as well. So this is kind of driving home that point that double-stranded RNA in the cytoplasm increases the interferon response, the antiviral innate immune response, then activates checkpoint presentation. And so now, we did this we did put this in the mouse brain, and basically you can see the survival rate is increased. This is very preliminary data. We actually repeated this experiment recently, thanks to Deepa and Jalisa and Ray, who's also a student in our lab. But basically, they show now that we're almost curing some mice with this knockout plus PD-L1 treatment, and you can see when you take these mouse brain and you do multiplex ELISA, you basically can see a very different expression of these cytokines in the knockout plus PD-L1 group. And so this is important because now you're kind of changing the tumor microenvironment significantly by knocking out this protein and potentiating immune checkpoint inhibition. So this is kind of interesting preliminary work. And so we saw this in patients. This is a patient who had before immune checkpoint inhibition and then after immune checkpoint inhibition. This was a tumor response. This is all kind of lymphocyte infiltration, and this is a tumor that has all active. And so the question here was, what is the double-stranded RNA pathway in this patient versus this patient? And so if you take the sample, this is the tumor pre-therapy, pre-immune response. There's really, green is supposed to be the double-stranded RNA. There's not much expression here and very low lymphocytic infiltration. But then after you go through immune checkpoint inhibition, this is a patient who had a response. You can see much higher double-stranded RNA pathways in this patient and much higher PD-L1 expression. So there's a correlation between this double-stranded RNA sensing pathway and PD-L1 expression in patient tumors that may predict outcomes. And so I'm trying to finish up this project a little bit to kind of show that as well. The last thing I'll talk about, which is the last 10 minutes, is basically retroviral mediated gene therapy. So I talked a little bit about oncogenic retrovirus. They're oncogenic. I talked a little bit about potentiating immunotherapy. Now I'm going to talk about retroviruses as a gene therapy. So this is work that I did when I was a resident, kind of looking at this one retroviral gene therapy. It's called DB107 now, but before it was called TOCA 511. And this data was studied in phase 1, 2, and 3 trials, actually. As Timur mentioned, again, this trial failed. So in phase 3 studies, this trial did not work out. So I've been interested. I said, well, there's a reason why this trial failed. What were the reasons why it failed? Can we do something differently to improve outcomes in these patients? And so this is a retroviral gene therapy that basically includes a transgene for cytosine deaminase. If anybody remembers, this is a gene that basically converts 5-fluorocytosine. 5-fluorocytosine is a antifungal. It converts it to 5-fluorouracil in the tumor cells, and that becomes an intracellular chemotherapeutic. And so the outcomes in the phase 1 trial were improved. So overall survival was 13 months in recurrent GBM compared to 7 months in matched cohorts. And so these patients had a lot more lymphocytic infiltration. This was published in Science Translational Medicine. And in the phase 3 study, they didn't really have much of a signal, between controls and when they randomized this in this double blind, there was no difference in survival. But a company, de novo, was able to acquire this data, and they did a novel biomarker analysis, and they figured out patients with this DGM7 biomarker had a much higher improvement survival compared to patients who did not have it. And so I said, what if we do this retroviral gene therapy just in patients with this biomarker? And so I said, that sounds, like a possible option. And this biomarker is really highly expressed in our South Florida population, especially in minority groups. And so the question is, can we use this biomarker to predict and do a kind of a phase 2 study looking at outcomes with patients with this biomarker and doing this retroviral gene therapy? And so that's what we're starting here at UM. Again, we're going to hopefully go to the FDA in the next 2 weeks. But essentially, this is a surgical resection and viral delivery trial. So patients with recurrent GBM who have this biomarker, you resect the tumor, and then you inject the virus using convection-enhanced delivery into the tumor bed. And the benefit in this trial is it's adaptive virotherapy. So we're doing repetitive injections. So if a tumor recurs, you inject again. And if a tumor occurs again, you inject again. And this is really important because there was a paper published just last year showing repetitive injections in recurrent GBM improve survival 4 times compared to a standard of care. So from 6 months to 24 months with just repetitive injections. And so now we're hopefully going to try to recapitulate that in this trial. And we're using convection enhanced delivery. The first trial was using hand injections, and we know that doesn't really work well. So we're hoping we can do this, and this was funded through the NIH. We're just lucky to get our first R21 with this clinical trial for recurrent GBM, me and Dr. De La Fuente. So the important thing here is we're using convection enhanced delivery for gene therapy here rather than doing injections themselves, because hand injections, unfortunately, do not work. And so, I'll kind of finish there with 15 minutes early. I know I talk pretty fast, but I want to thank everybody in the lab. Deepa, who's our amazing postdoc, came last year from urology, and she learned everything there is about neurosurgery and neuro-oncology in 1 year, and she's been doing amazing. A few MD PhDs, Christian, Anna, and Ray, who are all in our lab, focusing on different projects. They've all been amazing. A few medical students, Minor, who's applying to neurosurgery this year. Jay is another student who's from FIU, who's applying neurosurgery. Vedya has been a rock, other than now he's an intern, so our lab has slowed down a little bit. And a few other medical students. And thanks for all the funding from the NIH and Sylvester for our lab as well. So it's kind of a nice kind of turf war almost. The neuro-oncologists are kind of the gatekeepers of the clinical trial. So, anytime you do a clinical trial, you should always have the neuro-oncologists on board. So they're kind of also helping persuade the patients to go on your trials. And you want to have a trial that, takes away, they can compete with each other. So if you notice, these trials are very significant, very different. So one of them is unresectable glioblastoma that are getting laser thermal therapy. So this is a very unique niche. So these patients aren't trial candidates to begin with, so a lot of them have residual disease. They're in difficult basal ganglia location. They don't get a resection. They're just getting a biopsy and laser. So that's a very unique subset. The other trials is biomarkers. So they have to have this DGM7 biomarker, and that's only positive in like 30% of patients. So, that's going to, and they have to have some resectable disease, so that's going to be a very small subset as well. And then, then you have other basket trials for recurrent GBM. So for example, the radiation oncologists have another trial that's kind of a re-radiation study, and that can go for mostly all patients can get that study. And then, then there's other ones that are these big cohort studies from these cooperative groups. But, I think it's really important at UM that we focus on developing our trial portfolio at UM rather than using other people's kind of IP, because we are such a big center. We do see a large amount of patients, and so it's really important to kind of build that.
What percentage of your patients would you say of the population?
I think the big limitation is a lot of patients come from outside, and so they go back home to get their chemotherapy and radiation. I think Dr. De La Fuente does a very good job of recruiting as many patients as he possibly can. I would. These trials aren't up and running yet, but I don't know. It would be hard to say. I don't think the majority of them are on trials. Yeah.
Ashish, amazing talk. I think we were talking about how the residents really need to spend more time in the wet lab like this, and obviously you're starting a really phenomenal project here, just a year basically.
Thank you. I appreciate it.
You spoke about GBM as sort of like one group. I know you don't see it that way, one disease, but obviously the last 10 years has demonstrated a tremendous variety of phenotypes or genotypes. You didn't really go into that too much. Or are you developing like your own genotype of GBM that you're looking at? I mean, how does the sector, how does that influence, as Bart's point about how you do a clinical trial so you maximize it for success and not waste any money?
That's true. So in that Starlight trial, we're actually stratifying patients who have high HERV-K expression to begin with. So we're trying to figure out which patients have the highest expression, seeing whether or not our antiretrovirals benefit those patients the most. And that's going to be like a kind of a secondary outcome. We're validating that also with immunohistochemistry in the lab with the HERV-K protein. And in Vedya, it's actually done a lot of work looking at multi-omics and showing that there's, even aside from IDH mutation, aside from MGMT methylation, and aside from these Iveronic classifications, you can actually classify patients by the retroviral presentation. He's actually, he has this work he actually wrote half the paper already. We're just trying to validate some of the stuff in the lab, but essentially there's, we can almost discuss a retroviral classification system and whether or not that system could portend could portend a different prognosis with antiretrovirals. That's kind of our hypothesis, but we're a few years out. Yeah.
You produce the retrovirus and then you infect in situ, right?
Right. So the company actually has the IP from Tokagen, and so they're going to give us the retrovirus that they made a CLIA certified retrovirus, and so we're using their retrovirus and injecting it into the tumor bed. Yeah.
And have you tested the efficiency of the injection, the genetic injection?
Yeah. So you can see So this is a patient. This is a tumor sample that had been hand injected with this transgene, and this brown here is the cytosine deaminase. So this normally doesn't exist in cells, and you can see the brown is the immunohistochemistry for the transgene. So it does actually go and infect the tissue. The problem is you have to make sure the virus doesn't reflux out. And so that's the big issue, is that when you do hand injection, it will actually it'll reflux out. And ARIA actually published a nice paper on this in DIPG, where they did convection enhanced delivery, and they showed that this is actually going, when you do convection enhanced delivery of, they were using Pseudomonas exotoxin, but that actually showed that it actually went into the tumor bed through convection versus through not like through hand injection. So
That's a quick question. I saw that you use the CMV promoter.
yeah.
You test other or you don't think the CMV?
I think CMV is kind of a consistently active, very strong promoter, and so that's why they use the CMV. I don't think that they've changed the promoter for this specific one. I think for their other retrovirals elements they're trying at UCSF, I think they've changed it, but because it's such a strong promoter, I don't think they've found a reason to change it. Thanks. Help answer my question. Awesome. Yeah, we don't have much of a reason why this biomarker is working. And so this was just kind of post hoc analysis. And this is really, it's like a non-coding RNA, so it doesn't really make much sense. It's a gene, it's SHRB3. It doesn't. So I don't really have a good reason why. Why it's more efficacious. I think the big thing is what you're changing from your initial trial, and the things we're changing are, one is we're doing repetitive injections, because if your initial viral load is not good enough, then you have to kind of induce the immune system again with another injection. And the second thing we're doing is we're doing convection across the board. And they're doing hand injections before, and if you look at the hand injections just from people who did the trial, they were actually seeing that all the gadolinium was being refluxed out. So unless you're able to kind of do that, that's going to be the big change. Absolutely. Yeah, I agree.
Yeah, if you need to grab a coffee or something, go for it. I'm going to do a little review on nerve biology for about 5-10 minutes. So if you need a coffee, now's the time. I'm Shelby, and we'll be talking about peripheral nerve injury. And I wanted to focus on some of the data in clinical studies and what's out there now in clinical trials for nerve repair. Yeah, there is some lab funding from Integra, but not really relevant here. So what is the basic structure of the peripheral nerve anatomy? Well, it's filled with these cylindrical tubes, which are the within the internurium, the cylindrical basal lamina, and this provides the anchors for Schwann cells and axons. And this is really one of the magic of the structure of the nerve, and is really interesting and important to bear in mind when we think about methods of repair. This is sort of the train tracks that the axons grow down after a nerve injury, and after it's sutured back together or crushed, then the axons will travel down these basal lamina, and we'll talk about a little bit more. The intraneural anatomy is very complicated. This is a retrograde labeling of monkey median nerves from this guy, Tom Bouchard, I believe was out of Hopkins, orthopedic surgeon. And it just shows that along the length of the nerve from proximal to distal, the fascicular anatomy is not constant. So what was innervating the AIN proximally is now morphed sort of into a different position within the nerve as you travel distally. So that just shows you that these fascicles do not necessarily run in parallel, and that there's this intraneural plexus where the fascicles are kind of moving around as you travel down the length of the nerve, making it, again, more complicated as you try to suture back together two nerves. You really don't have any idea what's matching to what. So in nerve injury, there's several things that occur, one being electrical activity actually sent to the cell body, a deprivation of signals, and a retrograde transport of signaling molecules. All that becomes interesting as we look at ways to supplement repair. But after the axons are disrupted, they do want to travel down their their path, the path that they traveled before. And in a nerve crush injury, they will really reliably travel back down their basal lamina and back into their innervated targets. So typically in a crush model, the results of recovery will be good, especially in animals. As we move to humans, you'll see, and I'll mention it, there are several other factors at play, including distance away from the injury site to the target. When there's a suture line, that means the basal lamina are disrupted, and now we have appropriate and inappropriate targets being innervated. And you can see that trying to cross a suture line, these axons sort of get confused. They can even travel back to a U, as you can see one doing here, where this is one of the difficulties in suturing nerves back together, is that, as you can see, that there's a lot of disruption in terms of where the axons actually grow to. So now we've talked about proximal, we've talked about injury site, what's happening distal. Distally, the Schwann cells are deinnervated, they re-enter the cell cycle, they migrate, participate in clearing myelin and axonal debris, they proliferate, and they're associated with growth factor signaling. Mullerian degeneration is also occurring distally. And interestingly, there is this latent period for 1 to 2 days where the nerve is electrically intact. You can still, if you stimulate the nerve distal to the injury, you can still fire the muscle for 1 to 2 days until sort of the breakdown occurs via the Schwann cells. So back to the regeneration, this is another limitation in nerve, is that the rate of growth is about 1 millimeter a day or 1 inch a month. So that's a little something you can remember when you're thinking about these nerve injuries in patients. How fast does the nerve grow? Well, it's pretty simple, 1 millimeter a day, 1 inch a month. And that does change as we introduce suture lines, which can add an additional month to cross each suture line potentially, maybe 3 to 4 weeks. And this is obviously different in humans and animals, humans taking a bit longer than rats to cross suture lines. What are the clinical factors? Well, we also want to give a shout out to patient age here, because in pediatrics, the kids tend to do a lot better. Nerve repairing kids often can have really excellent results, and the older we go out, the harder it is to have any meaningful clinical recovery. Another one is the complexity of the nerve. We looked at this complex fascicular anatomy of the median nerve in primates, and we saw how when you travel from proximal to distal, the axons that were innervating one thing move into different parts of the nerve. Where the polar opposite of that is the spinal accessory nerve, where it really it's just a motor nerve. It's innervating trap and sternocleidomastoid, and patients even out to a year can have really excellent recovery. Well, that may be a little biased. 78% of patients recover full shoulder abduction. Obviously, the shoulder is abducted by several other muscles, so I don't know if that's confounded at all. But the bottom line is, if you repair the spinal accessory nerve, you have a good chance of recovery, as opposed to facial nerve. We've all seen these patients with facial nerve injuries. Why don't they recover as well? Well, because the facial nerve has to innervate 17 muscles, and even if you suture it end-to-end repair, it's cut distally, and ENT sutures it back together. They have an 83% of House-Brackman 3 recovery, but there's a tremendous amount of synkinesis, and the patients don't have good control of that side of the face typically. And that obviously goes way down with the graft repair, and that just speaks to the complexity of the nerve. So applying our understanding, and this again is just a little review here. We talk about the Seddon and the Sunderland classification. Seddon 1 is pretty simple. Neuropraxia, axonotomesis, neurotomesis. Neurotomesis is the always surgical realm. And we'll talk about sort of 3 different types of injuries. Does anyone know the rule of 2s or rule of 3s, Cater? Sort of timing in nerve repair. So how long do you usually wait on a clean transection? Yeah, up to 2 to 3 days. So then in a blunt transection, that's like a 2 to 3 weeks. And then in an injury and continuity, often you're waiting a few months because you really don't know how much recovery the patient's going to get on their own, and you rely on EMG nerve conduction study. But if there's any penetrating injury, really, it probably warrants exploration, except the bullet is a bit controversial. There's some people who argue early exploration on bullet injuries, but the traditional thinking is that as the bullet penetrates the tissue, it's creating so much disruption that it creates these sort of like neuromas, bulbs on the two nerve ends that will ultimately need to be trimmed back. And that's why people would wait 2 to 3 weeks to go in and do it. As opposed to a knife wound, you see those patients in Ryder and they've got to go basically to the OR in the next day or two. You really don't even have to wait any time at all. You can go directly to the OR. This is the boat propeller gunshot, and then this is the more complicated one where you're going to send, ACC clinic, get the nerve conduction studies, the typical stuff we see. So techniques in nerve repair, neurorrhaphy is that surgical repair of the two severed nerve ends. We need to identify the normal nerve, mobilize the proximal and distal ends, and avoid tension. They're usually brought together by sutures. There are some studies looking at just the use of fibrin glue, but I think that, we've seen patients where they mobilize and then the nerve ends rip apart. And I imagine that just fibrin glue is probably going to put undue stress on those two nerve ends. There's going to be a little bit of motion there. So unless the patient's fully immobilized, it makes sense to put sutures on. And additionally, we've been doing where we'll put a conduit over the suture end and suture the conduit to the nerve as well, which I think overall reduces the tension on the repair. But it has to be something permeable so that you can still have like nutrients permeate into the repair site. We're typically just doing the epineurial sutures. No one that I'm aware of really does these perineural or intravesicular sutures. I mean, I think it's happening in some places, but we don't really do that. And I'm usually using an 8-0 nylon vascular background. Some people go up to 10-0s. You can kind of strip some of the fibroblastic material off of the epineurium and then suture it together, or just suture the epineurium, as you see it. This stuff is, and so basically they looked at these different types of repair a long time ago and really found no benefit to doing a more intricate fascicular repair, and potentially that has some downsides of maybe introducing more scarring. So no clear benefit on that. And so now we get to this really challenging area of nerve injury with gap. Unfortunately, it's pretty rare. You're looking at the gunshot wounds, the boat propellers, the severe nerve stretch injuries to the arm or leg, where you actually have to resect a portion of the nerve, and now you're left with these two nerve ends. So what are we doing? And I mean, Adam, what the typical thing that we're doing in these cases is. Where are we getting all this nerve here? Yeah, exactly. We're grafting the sural nerve. And as you can imagine, you can see there's like a tremendous discrepancy in the size between the sciatic and the sural. This is a sciatic nerve in a young woman with a boat propeller injury. And so this has been a push for the last, I'm guessing 70 years to try to find other methods of repairing nerves besides going in and harvesting sural nerve. And I think the hand surgeons drive a lot of this literature. As you'll see, a lot of the conduit literature comes from hand surgery. And it's really different when you think about the small nerves and the big nerves, because the nerves that the hand surgeons look at are just small sensory nerves in the hand, not to minimize that, but they have sort of a different recovery profile. And I'll talk about that. So we know that there's tremendous advantage for using autograft because you introduce Schwann cells, but there are disadvantages, i.e. time and donor morbidity. We talked about the size match there. We have the basal lamina within the actual autograft, which is a huge benefit as well. We talked about this where basically at a certain point you'll run out of sural nerve, especially if you have a large sciatic nerve injury. And so this is sort of what borne out the trial that we'll talk about in a little. Here you can see another example of that. This is an injury to the peroneal nerve, 7-8 centimeters, where you basically got 2 incisions on the leg this big. Now you can harvest the sural through small stab incisions. We've done that before, but in either case, you're doing a lot of extra surgery. So what are the strategies people are looking at to enhance nerve? I've talked to you about sort of the challenges there. So what are we doing to overcome some of those challenges? You've got conduits, which we'll talk about. These have been looked at for a really long time, including just using an empty vein as a conduit. That has been sort of one of the originals. Cell therapy, electrical stimulation is one that's moving into clinical trials. Nerve transfer is one that we'll talk about. And then there's not really been any major promising drugs. Riluzole is one that's being investigated. And then differential repair techniques we talked about with the use of fibrin glue, epoxy, and others. So conduits in clinical studies, the bioengineered nerve graft substitute, these are called several things: conduits, tubes, axon guidance channels, all kinds of different names. The problem with these typically is that the Schwann cells have to grow into the conduit before myelination can occur, and they have really a limited migratory capacity, the Schwann cells. So in a long gap, an empty conduit is probably not going to work very well. But that has been looked at, and this is another primate study by Bouchard. I believe it was Bouchard that looked at basically autograft or conduit at different lengths of gap, and as they increase the gap length, the conduit starts to fail, basically. And that sort of just speaks to what we mentioned about the migratory capacity of Schwann cells. So lots of studies that have looked at this, and as you look to the right, you can see their conclusions that basically they're either comparable or slightly worse. Here's one study that highlights it pretty well. This was from 2018. And this is a collagen conduit filled with collagen filament. So now you're trying to recapitulate that basal lamina. You need to give the axons something to grow on. So if it's a hollow conduit, they're just going to grow around the walls of the conduit. But if you're introducing some structure into it, they can grow on that. So they used sensory nerve defect at the wrist or distal, so basically hand, and less than 30 millimeters, less than 3 centimeters, so something like that. Small defects in sensory nerves in the hand, basically. And here's sort of what that looks like intraoperatively, where they suture the nerve onto this small conduit there. And they had conduit group had 49 versus 38 patients, similar gap length of really small. So, 12 to 18 millimeter gap. And they show similar sensory recovery in both groups with no serious adverse effects. So this is sort of where a lot of the conduit literature comes from, is that these small nerves in the hand. So I think we just need to take that with a grain of salt, so to speak. Right. Longer outcomes here out to 2 years, prospective randomized trial looking at collagen conduits in a 6 millimeter or less in the forearm with no difference in outcomes in the two groups between grafting versus conduit. So in these types of nerve injuries, conduit seems very reasonable because you don't have to go and harvest sural nerve. And so this is a cool sort of synopsis from McKinnon and Zach Ray out in WashU. And they suggest that authors limit their use of nerve conduits to the repair of non-critical small diameter sensory nerves in gaps less than 3 centimeters. So they're really saying, for most of the stuff that neurosurgeons do, we shouldn't really be using it. But then the elephant in the room we need to talk about is the acellular nerve allograft, which is really used a lot by our colleagues in hand surgery. We do use this as well. I kind of put it in the same group as conduits. It's basically decellularized nerve from cadaver sources. And so you still have these basal lamina scaffolds, but again, the Schwann cells do have to migrate into it. It has the same handling characteristics as a nerve, which is beneficial, and it has all that basal lamina. But unfortunately, I think it should be treated in the same way that conduits are, in the sense that, long gaps, big nerves, we probably should limit the use of this. But we'll talk about it, and you'll see in the next coming slides that people are trying to supplement this with cells, and maybe that's something that we can do to try to overcome and that's another benefit to conduits that we'll talk about. There are lots of literature on these. It's called Avance by Axogen. It's got a big piece of the market in peripheral nerve injury. So that's why I say it's elephant in the room, because, without talking about it, you're not doing any justice to this field, which uses it a lot. But I think it should kind of be treated in our minds like conduits in a sense. So here's some of the literature on that. I just put there at the bottom. So let's talk about cell therapies, because there are several clinical trials, including one here at on using cells to supplement nerve repair. So what I mentioned before is axon guidance channels provide this nice ability to introduce cells at the repair site. Schwann cells are the natural choice, but there are several other stem cell types that we can look at. I also want to give a quick historical note. There has been in the past research into the use of cellular allograft. So big nerve injuries, actually getting live donor allograft and immunosuppressing the patient. This has been studied and published in 2001, but it does require up to like 3 years of immunosuppression. So it's probably not reasonable. And you can even have rejections, which they did in one of their 7 cases. They they use small diameter nerve grafts as their donor, but most of the nerves injured were very large. Here's a couple examples of this. Huge injuries. So they were sort of these compassionate use exemptions, sort of FDA probably granted them, based on a case-to-case basis, I bet, or possibly a clinical trial for these really devastating nerve injuries, 22 centimeters. So it's an interesting option and something to think about when you think about sort of a historical context. What are the cell trials ongoing now? Here's one we found in Poland, intraoperative ADSC administration during nerve release. So it's revision neurolysis, and they're giving adipose-derived mesenchymal stem cells onto the actual fascicles. So they micro-inject adipose-derived mesenchymal stem cells. This study is still recruiting. Last update was in 2020, so we'll see what they find out of Poland. Here's one out of Indonesia looking at, this one's a little complicated. They're basically looking at human amniotic membrane with allogenic adipose-derived mesenchymal stem cells for augmentation of nerve transfer. So the nerve transfer, and then they're wrapping it with the human-derived amniotic membrane, and then they're adding the allogenic adipose-derived mesenchymal stem cells. So they're really throwing the kitchen sink at these brachial plexus injuries. So this one will probably be a little bit hard to interpret, but kudos to those docs out there that are studying that. This is one here in the US, evaluating treatment with Avance, the nerve graft that I talked about, with autologous bone marrow derived aspirate for the repair of peripheral nerve injuries up to 7 centimeters in length. And I believe this is ongoing. I believe they've recruited I think they've recruited 15 subjects. I think this is based out of Texas. So this is just what we're talking about, adding bone marrow derived autologous stem cells. They're not doing any, I don't imagine they're treating these very much. They're probably just taking a bone marrow aspirate and putting it onto the Avance grafts in the hopes that this introduces sort of those progenitor cells, which can differentiate into Schwann cells. Again, it's not very targeted, but it's nice to see that some of these, and hopefully one of them will click.
Very unlikely that a bone marrow stem cell will turn into a Schwann cell. But
Yeah, but they are, yeah. So here's another one, Mansoura University, investigating platelet-rich plasma to treat myocarpal tunnels. I mean, this is sort of on the periphery of what we're talking about here, but it's interesting. Platelet-rich plasma treatment versus steroid injection. So this one doesn't even require surgical intervention. It's just injected right into the carpal tunnel. And then we have our study, which is registered at clinicaltrials.gov. Dr. Levy's a PI, with the use of autologous human Schwann cells in peripheral nerve repair. And we talked about the benefits of Schwann cells, what they do, helping clear myelin debris required for axonal outgrowth. Can we just add them to the repair site and thereby enhance recovery? Several patients have been enrolled prior to the initiation of the study, and now as the new iteration of the study is ongoing, the first patient was enrolled a couple months ago, brachial plexus injury. And the way it works, I've been up here talking about this before. I'm sure you've seen this slide before, but essentially there's the repair ongoing. There are the small cable grafts that we've harvested from the sural, and then a microinjection of autologous Schwann cells, which are grown in the lab, is then injected into the repair site. And those autologous Schwann cells are harvested 2-3 weeks prior from a sural nerve biopsy, taking a piece of sural out in a separate procedure. And then at the time of definitive repair, injecting the Schwann cells into this. So if you see, in Ryder, these patients that are gunshot wounds or motorcycle accidents, just be thinking about this as a possible option for them. We're almost approved at Jackson, I believe. So, and then The other thing I wanted to mention is this can't be like in a clean transection where you're going to be repairing it right away. It really needs to be one of those either in continuity or blunt transection so that you have a little time for the Schwann cells to grow. And it has to be a gap of greater than 5 centimeters.
Make sure that it doesn't like stay in there,
Well, they have like a limited migratory capacity and there's tons of data from the Miami project showing that they don't generate tumors. So I think by wrapping it with the collagen wrap and injecting some fibrin glue that most likely they do. We talked about, is there any way to actually trace them with like MR tracing? But besides one of the patients, postmortem study, I don't imagine that there's a good way to evaluate it.
I think that's a great question, and that's what the fibrin glue is important to, kind of cap the ends of the tube, so to speak, otherwise that it would run out the sides. But it's again something that Dr. Burks is working on. It's not the greatest delivery system, and we could do a lot better and possibly use, nerve inclusion to deliver the cell.
Yeah, the next generation conduit is sort of this is that paper looking at that sort of next generation conduit, in animals, showing that it is possible to deliver cells through this off of the back of this paper, which showed that cells could be delivered in rats through an empty conduit from Dr. Levy and Yurko Barakol back in 2013. So this is sort of where we're trying to go. Right now, it's just basically hoping to show that there's safety in injecting the Schwann cells into the repair site like I showed you here. So yeah.
Could you do a gallon of injection like while you're injecting? I can look at MRI like right after the repair. I mean, it's just stay there for an hour or two. I guess it confirm.
Yeah.
The label, so that would all you see is start.
Oh, wait. Yeah.
What's that thought? We'll probably see the gadolinium leaking everywhere and then we'll say, oh, great, we should. So there are disadvantages like we talked about to the use of Schwann cells. Is there another option? This is another thing that we're looking at now. We've got two postdocs from the lab here, Emily and Erica, that are going to be investigating, the use of exosomes or small vesicles from the cells and trying to use those instead of Schwann cells. I don't imagine we're kind of getting the results of that data now, and they probably don't have as robust an effect as cells, but they have very little cost to be able to add them to a repair site. It doesn't require any immune suppression, and you can sort of have a universal donor of those. So hopefully. So I'm going to change gears and talk about electrical stimulation now, and this is something that's being investigated. We talked about, at the time of injury, there is an electrical stimulus that goes to the cell body to sort of reactivate it, get it ramped up for recovery. And people have realized that by stimulating the proximal nerve stump after injury, that they can sort of harness that even further. So it's mediated by this mechanism that's been relatively well worked out. There's been so many preclinical studies in this that they've been able to figure it out. And it dates back to experience from the 1950s. At what level of stimulation and for how long is really the question, though. And that's sort of what's, a lot of this literature you'll see comes from Alberta, Canada. Tessa Gordon has led to this big push in this type of research. And I think right now what it is after repair, 1 hour stimulation. That tends to be the typical protocol, but people have looked at prolonged stimulation. As you go out past 2 days or something like that, you start to see a deleterious effect. Here in 2000, authors showed that electrical stimulation via wire wrapped around the nerve stump proximal to the repair site increased motor axonal recovery, regardless of the duration, 1 hour versus 2 weeks in the rat femoral nerve. And rats may be a little bit more tolerant of this than humans. So here's some of the clinical studies. This one was published in Experimental Neurology in 2010 from that Tessa Gordon group. They showed that chronic compression in the carpal tunnel patients after 1 hour of stimulation after surgery, they had enhanced motor recovery at 6 to 8 months post-op and at 12 months post-op. So that was sort of one of their original publications. Then now in 2015, they're looking at digital nerve transection and repair 18 patients in each group, and they randomized them to 1 hour stim versus sham. And electrical stim had better sensory recovery, and the questionnaire data trended better. Here's another sort of a little bit weird study, but it was in 2018 ENT where they did intraoperative stimulation of the spinal accessory nerve, showing reduction in deficits after surgery. Only 25% experience, 25% of functional loss seen in the control group. Here's another from 2020, again, using a severe cubital tunnel syndrome, 1 hour of electrical stim postoperatively, 11 versus 20 patients with 3-year follow-up, showing that electrical stimulation did enhance hand recovery and pinch grip strength. So animal data supports it, and it may have a role in preconditioning prior to resection of a nerve tumor, for example. There are several clinical studies looking at this. This is one funded by Checkpoint, which I don't know if you've seen it in the OR. It looks like a small pin. It's a monostimulator. It's called the Checkpoint Nerve Stimulator, and they're trying to enroll 100 patients. I think they have 3 or 4 sites. I imagine it's either plastics or orthopedics doing it. And that study has just sort of kicked off, looking at cubital tunnel repair, stimulating the nerve intraoperatively using the Checkpoint. Here's another one out of Alberta where, again, a lot of this comes from with electrical stimulation, improved hand function, nerve regeneration. This one, placebo controlled. Again, they're looking at 1 hour versus sham. That study is ongoing. Here's one in sick kids, a little bit of a complicated study looking at facial nerve palsy patients with cross facial nerve graft with electrical stim or sham. So they're basically applying it to every type of nerve injury model. Probably does have a beneficial effect based on all the animal literature and some of the early clinical literature. This one's enrolling a small group of patients. Another one here, Regain study out of Alberta, 110 participants. This one is new, just starting to enroll, and they're looking at a novel device that they can place into the repair site. So that's sort of where this is headed. I know Zach Ray at WashU is working on a device. Potentially in the future, there'll be nerve repaired. A small device is left on the nerve, like a collar or something, and then it can be removed a few hours post-op so the patient can wake up. Imagine it would be uncomfortable, though, to receive the nerve stimulation, so maybe they'll have to have a regional block or something like that. I'm not sure how they're doing it. We'll talk a little bit about when repair is not an option. We can talk about nerve transfers and also TMR or RPNI, which is something that's being actively looked into at this time for stump neuroma pain. So the incidence of painful neuroma after amputation ranges from 50 to 80%. So the majority of patients after amputation will have a degree of stump neuroma pain. So now we're looking at targeted muscle reinnervation for this problem. There's been tons of sort of avenues that people have looked at for neuromas. And unfortunately, unless you give that nerve a target to grow into, it's just going to grow into another scar ball wherever you put it. So by capping it or cutting off the neuroma, you're really just delaying the inevitable. These patients will redevelop their neuropathic pain until you can try to give the nerve a target. And I think this comes from the plastic surgeons. And what this was originally looking at was, and orthopedic surgeons, for smart prosthetics. They were putting the stump neuromas into some of the pectoral muscles and using that to control a smart prosthetic. And they realized that these patients had less neuropathic pain. And this publications came from, starting in the early 2010s, showing that in the TMR patients, they had very good resolution of their neuropathic pain. So that's led to sort of the development of this for neuromas. And actually people are looking at doing TMR prophylactically after amputation, then taking those nerves that are left, plugging them into muscles so that patients don't develop as much neuropathic pain. There's at least 4 actively enrolling clinical studies. I think probably more that are looking at prophylactic TMR. There's another one called RPNI, where basically you just take a piece of free muscle and just wrap it around the nerve stump or nerve end, and that hopefully gives it something to grow into. I think the TMR is sort of the gold standard. This is one that's being investigated. It does show good results, but I imagine that for that muscle to survive, it needs to be in a well vascularized area. So if vascularity is a concern, it's probably unlikely that that small stump of muscle or that small piece of muscle is actually going to be surviving. And here's an example of RPNI. So, and then I'm going to again switch gears to talk about nerve transfer. And this isn't exactly for nerve injury, but this study is nerve transfer to improve upper extremity function and quality of life in patients with tetraplegia. And this is initiated by Zach Ray at WashU, and we've subsequently joined this group. And so what it relies on is the fact that the below the site of injury, that the motor end plate is intact in continuity, so that we can leverage that to then transfer from proximal to the site of injury to distal to the site of injury. So some of the typical transfers we're doing are branch of the axillary nerve to the radial nerve for tricep function, because most of these patients have basically deltoid and bicep. That's like the typical, C5, C6, somewhere in there. These are the ones that really benefit from this transfer. And it's that, again, axillary to radial, musculocutaneous to AIN, because there's redundancy in elbow flexion with both bicep and brachialis. So we take a branch of the brachialis, plug that into the median nerve to try to innervate the hand, flexors of the hand, and then using the supinator branch of the radial into the PIN branch of the radial to try to give them finger extension as well. And even if they have some wrist extension, we're still able to do this because ECRB innervates some of that wrist extension, and we're going sort of distal to that, so we're not injuring the wrist extension that they do have already. And so this is ongoing at 12 centers. You can see here it's headed by WashU. We're on there, and we're actually going to be moving to also include high tetraplegia patients from C1 to 4 injuries. We've right now enrolled one patient here, and we're hoping to have another one enrolled in the next couple of weeks.
I'm glad you brought that up. I mean, that's a fascinating case, and since some of these people will be taking care of the patient, why don't you tell us about the patient?
Yeah, there's a guy, Burgi, who did a neurocritical care fellowship here. He's over at LSU New Orleans, and he treated a federal agent who basically had a fracture dislocation at C5-6, has a complete injury at C6 with a cord transection on MRI. So he has, I'm not sure his exam, we haven't met him yet, but he has bicep function, deltoid are definitely intact. And then below that, really nothing, maybe a little bit of wrist extension. He has a cord transection though on the MRI. So we're hoping he was treated last Wednesday. So we're hoping to enroll him in this study where we're actually going to do preemptive nerve transfers, just knowing that he's not going to get any better with that. So this is to try to give them hand function. And what they've shown out of WashU is that at, basically a year, 2 years out, that patients are starting to get grasp function. So before, when they just had, the biceps working, now they can start to use their hands a bit more and, power the wheelchair and hopefully grasp things or, feed, drink, and that kind of thing.
My only question is, in the study, they must have very specific inclusion criteria and exclusion criteria. And so what is the earliest time to enroll? Because everybody, the study is for chronic patients. So
Yeah.
Would this be someone who's not meeting the criteria, or?
This would not be meeting the criteria. The typical thing is like 3 months, 4 months out, being the earliest. So I talked to Zach, and this would need approval from the FDA before enrollment. So that is why we're hoping to transfer him over to rehab and then.
Yeah.
Oh, sorry. Yeah. The DOD, and the FDA.
Is
Yeah.
there an advantage in weight?
Yeah. The thing about waiting is as we see the typical, we don't want to treat someone too early when they were going to recover some hand function, or they were going to recover some of those muscles that we're taking, because it does require you to cut nerves in order to do those end-to-end transfers. So that's essentially why you would wait. But if you had that transection, then presumably we don't need to wait. That would be the thought process.
The only thing, and I think about getting to that point is that, you know, everybody improves, right? Everybody with a spinal cord injury, whether it's a transection or, and I mean, they usually improve one level. And, you know, even AKA, so does that impact, you know, the thinking here in terms of the actual nerve transfer strategy? Again, I think the point to understand is C6, and then you might become a C7 with time. It's probably something that needs. I've never seen the MRI. I just saw the CT. I know the cord is very badly injured, but we know that people improve one level usually AKA subsides in the cord.
Yeah, no, that's a good point. I think I was hoping to wait maybe 2 to 4 weeks at least and see how he does at that time. Probably 3-4 months would be ideal, and you really see where he is exactly. But on the MRI, there is complete discontinuity. You can actually see the two ends of the cord, and the canal was basically totally cut. So in that sense, but no, I understand that point. So yeah, well, it'll be a little bit of a moving target. Worst case scenario, it's not approved. We'll treat him outside the study in about a month time from the injury. So,
And so it'll be coming here and then going to our rehab center as well.
yep, exactly. Yeah, so that's basically everything I have. We'll give a shout out to everyone here. I had Eric on here on the newest iteration of the talk, but welcome to Erica, our postdoc joining us. Emily and Joe, thanks, and Meredith, thanks for the help in sending me over some slides here. And to try to compete with Ashish, we do have our own Instagram as well. So you can find it there.
So coincidentally, I saw one of your patients. And, you know, one of the things is, when you do a transplant, how much time is connection?
Nerve transfer?
Yes. What are, do you have a protocol to protect that area? In other words, you send patients out of the operating room not with, you know, some sort of a shield device protection, because as you know, quadriplegics are lifted, bent, their arms are manipulated, moved around. And this is a very delicate situation where the nerve is in their trophic arms is very superficial. And, you know, I just wonder what your protocol is for protecting your work for a period of time from external trauma, from, you know, type of movements, from.
Yeah. I mean, in that patient, yeah, no, we don't have a good protocol, honestly. I mean, I'm just telling them to wait about 2 weeks before initiating therapy, and then just to sort of ease their way back into it over the course of 6 weeks. And hoping that, you know, they're not really going out to the extremes of their mobility, hopefully. But yeah, I mean, I think that's something we need. I think that's a good point. I need to basically have that drawn up.
Protective device to protect the area for a period of time. You know, just because of the nature of how they're handled, they're lifted, pulled, or moved all over the place. It's such a delicate transfer.
True. Yeah, you think about it that way when they're being lifted out of the chair. Yeah.
The people handling them aren't educated on what somebody needs. It's very delicate surgery.
Yeah, and I just didn't want to limit too much their ability to be transferred and that kind of stuff, because I know that that's sort of like vital to their day-to-day life. So that's where I was sort of having I didn't want to limit them too much also, but I understand what you're saying.
For the residents, fellows, medical students, I got this email from this group who's demonstrating a robot on the 12th floor, 1270 of the CRB, and it's specifically for nerve repair. I don't know really the details, but it's basically to help with microsuturing and, you know, 12Os and nerve transfers and stuff. They had a paper along with it. I have no idea what it's about, but they're here for 2 days today and tomorrow. So if you go to room 1270 in the CRV, you can see what it's about, maybe try it and things like that.
I based on the picture, well, we'll see what it looks like. I just saw the picture of it where it has it's a robot with multiple arms and it's used to do micro suturing, so.
And then the other thing is Dr. Stark, if you could make it, is receiving an award today from the dean's office for his research. And they have the faculty awards every year, and he was named the best researcher at the university.
Wow.
Also in the CRP at 6 o'clock. Free alcohol and whatever you want. Awesome, thank you,
Cool.
Shelby.
Thanks.